Okay, so now we're going to talk about protein metabolism. And in this first lecture, this overview, we're going to talk about the genetic code. We'll talk about how it's redundant, how it is mutant resistant, and then we'll talk about how it's a little bit flexible with respect to what is called the wobble effect. We'll also discuss the structure of the transfer RNA and how it's important, of course, in building our amino acids into our proteins. So the genetic code is basically going to translate the message from our DNA that was packaged into our messenger RNA into something useful that the gene or your cells can use. And so with respect to protein synthesis, there were three major advances. One was, of course, the identification of ribosomes as the site of protein synthesis. And then we also have the formation of your transfer RNAs that are going to be activated. And these are going to be catalyzed by amino-acyl tRNA synthases. And then finally, this just concept, the 4-letter language of DNA being translated into the 20-letter language of proteins. And so basically, with respect to each strand of DNA or a messenger RNA, the sequence is going to have 3 possible reading frames because our codons are in 3s. And so each reading frame is going to give a different sequence, but only one of the sequences is going to give a protein. So you can see basically this shifting of the sequences, just basically shifting your your codon by 1 or by 2, and that will shift the actual reading frame of the codon. And so it is known that the code is read in a sequential manner starting at the 5' end, and it goes to the 3' end of the messenger RNA. The codon is a triplicate of nucleotides, so we have a triplicate. And these are, of course, of our nucleotides, and this is going to encode for a specific amino acid. And then the reading frame is going to be that translation of these codons, and then your first codon is going to be what starts the reading frame. And so it's important, again, to make sure that the reading frame is, in fact, the frame that will give you the functional protein. Now, the code is said to be degenerate because an amino acid can be specified by more than one codon. And that is shown here in this table, in which you have methionine shown here, which is, of course, the start codon. It has only one codon, whereas you have serine over here that has 6 codons. So different amino acids have a different number of codons that are available. Important to note that each codon only specifies one amino acid. And there are 64 possible codons. Now you can think of the codons as a dictionary, and this dictionary with respect to the amino acid code are basically written in the messenger RNA. You're going to always have our methionine, which will be the initiation codon, and then there are 3 codons that are stop codons. And so those are, of course, shown here, the stop codons. And our methionine, our start codon, is shown here. And so with respect to our open reading frames, we're going to have a stop codon that occurs every 20 amino acids in a reading frame. A reading frame without a stop and 50 amino acids is an open reading frame. And so long open reading frames are usually the areas where there are genes, and these genes are going to encode for a protein. So when you're going to look at the DNA for genes, you're going to focus on long open reading frames. Now let's talk about the transfer RNA. We've already seen this molecule already, of course. They are going to have a 5' terminal end, and this is going to have a phosphate. They are going to have a 7 base pair stem that includes a 5' terminus, and this is going to be the acceptor. And they're going to have a D loop and this funny T loop, and these are both going to be for structure. And they're going to end with a CCA ending at the 3' end. Now D is going to be for dihydrouridine. And then, of course, you're going to have the amino acid arm, which is a specific amino acid that is going to be added to the arm. So here's our amino acid arm. In this case, we have methionine in that place. The anticodon arm is going to contain the anticodon, just shown here. And that's going to, of course, be opposite of what the messenger RNA codon is. The two D and T arms are going to be important in folding. And so what's important to understand is the anticodon is where the tRNA base pairs with the messenger RNA, and the first base in the codon, which the messenger RNA, remember, is 5' to 3'. In our codon, this is going to be in the opposite direction, similar to what would be in a double-stranded molecule. So think of it in that terms. So it's going to be 3' to 5'. So the first base in the codon will pair with the third base of the anticodon. So this is an anti-parallel alignment. Which is similar to our DNA molecules. So our transfer RNAs can recognize more than one codon, and this is an important concept. Because remember that base pair hypoxanthine, well, it can become inosine. And inosine can bond with either a uracil, a cytidine, or an adenine. And so this ability for the flexibility of inosine to bind with these 3 different amino acid, or these 3 different nucleic acid, is what Crick proposed would be what we call the wobble effect. And so if you look at your codon, you can see the first 2 bases of messenger RNA are always going to form a very strong Watson and Crick interaction. So those first 2 bases are going to correspond with the appropriate base. So here we have cytosine with the guanine, guanine with cytosine. Here we have cytosine, guanine, guanine, cytosine, etc. Now, what is important to understand is that the first base of the anticodon, which is going to bind with that third base of the actual codon, will determine the number of codons that are going to be recognized by that specific transfer RNA. And what that means is if you have a Watson-Crick base in there, if you're going to have an A and C in the first position, then those anticodons in the base pairing is very specific, and you can only have one base pair with that. However, if you have a U or G in the first position of the anticodon, the binding is going to be less specific, and when you have an inosine in the first position, 3 different codons can be recognized. So here we have inosine here, which is going to recognize A. Inosine here is going to bind to uracil, and inosine here is going to bind to cytidine. So an amino acid is specified by different codons. The codons that differ in either of the two first bases are required by different transfer RNAs. So there are a minimum of 32 transfer RNAs that are required to translate all 61 possible codons. So we have 32 tRNAs needed. And then 31 are needed to encode for amino acids, and 1 for initiation, that methionine. So as we'll learn, that initial methionine that starts the initiation is different than the methionine that is actually in the protein, within the structure of the protein. All right, so the genetic code is also mutant resistant. So we have missense mutations. And these are where you have a single base pair replaces another. And so your third wobble position, in which you have a new base pair replaces another in the substitutions, are going to produce a change in the encoded amino acid only 25% of the time, and that's because it has flexibility in that position. Most changes are going to be silent mutations, where the nucleotide is different, but the encoded amino acid is the same. And that's because there is degeneracy in the code. And then a transition mutation is the most frequent of our missense mutations. And so remember, our mutations can lead to a hydrophobic amino acid changing to another hydrophobic amino acid. And so the organization of our genetic code is such that if there is a mutation, the likelihood that the amino acid would change to a similar amino acid is greater than amino acid changing to a very dissimilar, such as a hydrophilic for a hydrophobic, etc. There is also another concept of translational frameshifting. And the reason for transcriptional frameshifting is to allow two or more related but distinct proteins to be produced from a single transcript. Now, viruses use this a lot. So here we're actually showing this is HIV genome. And so you're showing a GAG reading frame. GAG is part of their envelope structure. And within the GAG reading frame, you have an overlap with their polymerase reading frame, which is shown here. And so what basically happens is in the GAG, you have the stop sequence, which will then produce GAG. But if you do a frame shift, you can start to produce your polymerase. And so most codes are going to be read through that stop codon. And again, the two other possible reading frames are usually not going to contain useful information. So when you change this codon to starting at the U, or you start at the A, those are typically not going to make a protein. Some genes are going to be structured so that the ribosomes have a hiccup, and this is the case here where you have a hiccup. And the ribosome is able to jump to the other reading frame. But again, most of this is done in viruses. Now, RNA are also going to be edited, and some messages are going to be edited before they're translated. And these are mostly going to be in mitochondrial chloroplasts. But RNA editing can also occur through adenosine or cytidine deamination, as we discussed already. And so if adenosine is deaminated by the adenosine deaminase, this is going to generate inosine, and inosine is going to be interpreted as guanosine during translation. Cytidine deamination is going to be caused by the ApoBec family proteins. It's ApoB messenger RNA editing catalytic peptide, and in cytidine, you're going to deaminate to uridine. ApoBec family proteins are important for antiviral. It's thought to be a normal antiviral way to introduce a mistake in the growing virus chain. So an example for RNA editing at the level of eukaryotes is that you have different ApoB and ApoB100, ApoB48. And this is for our lipoprotein particles. So lipoprotein particles. And those are the chylomicrons, the very low density lipoproteins, etc. So the ApoB100 is found in very low density lipoproteins. And the ApoB48 is found in our chylomicrons. And so it would make sense that our liver would generate the ApoB100 because that is the site where you generate your VLDLs, very low density lipoproteins. And it would make sense that the intestine would make those ApoB48s because that is where you make your chylomicrons after you eat. And so your intestine makes those proteins. And you can see that basically in the intestine, they have an insertion of a stop codon. So this is going to form a shorter, this is the ApoB48, and it's truncated or shorter. And then this is the ApoB100, which is the full length of the actual RNA reading frame. And so the deamination it is what occurred from cytidine to uridine in the intestine that caused this stop codon to be formed. And this converts glycine to a stop codon. And so again, you get a smaller truncated protein in the intestine. So in summary, again, we have our degenerate genetic code. We have our amino acids that can be translated by different codons. Our codons are always in 3, so if you shift the frame of the reading frame, you can often make that a deleterious. It's not going to form a functional protein. We have our stop codons, those 3 stop codons. We have our start codon, which is that methionine codon. We have our wobble at that 3rd position of the anticodon, which could be an inosine, for example. And then frame shifting is a way to allow changes in your actual RNA transcription.